4.5 billion years. Let that sink in for a second.
Yeah.
I mean, that's the epic timescale we're talking about, right? This incredible journey from, well, a lifeless rock to the absolutely breathtaking complexity of everything alive today.
It really is mind-boggling when you think about it.
And that's exactly where we're diving in this time, the ultimate origin story.
Yeah.
Not just creatures and plants, but life itself, right down to the molecules that make it all tick.
Yeah, we've got this fascinating guide, let's call it Okyusei Ontchang, which kind of maps out this incredible path from the universe's beginning, early Earth, all the way to, our complex genomes.
It feels like a huge undertaking, trying to piece together how simple chemistry could spark something that replicates, evolves, becomes us, and everything else.
It is, and our mission here really is to pull out the most significant insights from this material for you. Try and make these, frankly, profound processes clear and engaging without drowning you in, jargon or excessive detail.
Okay, let's do it. So our guide kicks off right at the start, the cosmic timeline. We're talking the Big Bang, roughly 14 billion years ago.
Then things cool, gravity does its thing, and around 10 billion years ago, our Milky Way galaxy starts forming.
And then fast forward quite a bit, about 4.6 billion years ago, our solar system, including Earth, forms from all that swirling gas and dust.
And it's really important to picture early Earth. It was completely different from now. Our material points to a planet with this huge ocean, probably global.
Right.
And the atmosphere. Very little free oxygen. Instead, lots of methane, ammonia, water vapor, hydrogen, stuff like that.
So you imagine this primordial soup under a, well, pretty alien sky. Which brings us straight to the big question.
Yeah.
How did life's building blocks even show up in that environment?
Yeah, how do you get from simple molecules to something alive? Or at least on the path to life?
This is that mystery of abiogenesis, isn't it? Life from non-life.
Exactly. And the material we're looking at suggests the first key step was the emergence of basic biochemical systems right there in the early ocean.
So the idea is that life's ingredients could actually form spontaneously back then. It wasn't some, impossible miracle.
That seems to be the key insight, yeah. Plausible conditions could lead to these building blocks naturally. Less miracle, more. Inevitable chemistry, perhaps.
And it sounds like this first step happened relatively quickly, geologically speaking anyway. There's evidence for cellular life popping up around, 3.5 billion years ago?
That's right, tiny microfossils found in really ancient rocks, like in Australia. And they look remarkably like modern bacteria.
Wow. But, okay, our guide makes a point here. Cells, even simple bacteria, are already pretty complex. They probably weren't the absolute beginning. Right.
Exactly. Simpler systems must have come first. And the leading idea, highlighted in our sources, really zeroes in on self-replicating RNA molecules. They might have been the precursors to the first genomes.
Ah, the RNA world. So before DNA was king, RNA was maybe doing everything, carrying info and doing the work.
That's the hypothesis. But how do you even get RNA? How did its building blocks, the nucleotides, form on that early Earth?
Good question.
Well, this is where that classic experiment comes in Miller. Our material details it, 1952, right?
Yeah, I remember learning about that. They zapped some gases.
They simulated early Earth conditions, those gases we mentioned, plus electrical sparks for lightning. And remarkably, amino acids formed spontaneously. Glycine, alanine, valine, the building blocks of proteins.
That's huge. It showed the basic ingredients could just appear.
It was a massive proof of concept, but as our sources point out, making nucleotides, especially the ribonucleotides for RNA, is trickier. You need the sugar, ribose, plus the base all linked up correctly.
Yeah, that seems like a more complicated assembly job.
It did for a long time. But more recent research, which our guide mentions, suggests maybe there's a more direct route. Perhaps complete ribonucleotides could form directly from simpler chemicals readily available back then, things like cyanamide, cyanoacetylene, glycolaldehyde.
Ah, so you don't need to build the sugar and base separately and then link them? They might form all in one go?
Potentially, yes. Which definitely makes the whole RNA world idea seem, more plausible.
Okay, so let's say we have the ribonucleotides. How do they link up into chains, into RNA polymers?
Our material mentions a few possibilities. One idea is base stacking the flat nucleotide bases kind of stick together weakly, which might help them line up. And maybe stabilizing surfaces helped, like minerals in clay or maybe ice crystals, or even just evaporating pools concentrating the molecules.
And so physical environments could have played a role.
Absolutely. The exact mechanism isn't fully nailed down, but the point is there are plausible geochemical ways it could have happened. And importantly, similar processes might have formed polypeptides protein precursors too.
Right. Now, this brings us to that classic puzzle, the chicken and egg problem. DNA holds the blueprints, but you need protein enzymes to read and copy the DNA. But the instructions for the enzymes are in the DNA.
Precisely. It's a paradox. Which came first?
So how does the RNA world solve this?
The solution, as our guide explains really well, came with a revolutionary discovery: ribozymes. These are RNA molecules that can act as catalysts, like enzymes.
Uh-huh. So RNA isn't just storing info, it can actually do things too.
Exactly. It has this dual role. It can be the blueprint and the machine carrying information like DNA and catalyzing reactions like proteins. That neatly sidesteps the need for both complex systems to arise simultaneously.
Okay, that makes sense. And these ribozymes, they're not just theoretical.
Oh no, our sources give example. Natural ones found today can cut and paste other RNA molecules. And crucially, the ribosome, the protein-making factory in all our cells, has an RNA core that actually catalyzes the peptide bond formation.
Wow. So RNA is still doing catalytic work today.
Absolutely. And lab-made ribozymes. They've shown even more potential synthesizing nucleotides, even copying other RNA strands.
So you can imagine early replication in this RNA world. An RNA strand acts as a template, nucleotides line up.
And then maybe they spontaneously link up, or perhaps another ribozyme helps catalyze the linkage. But, as our guide points out, this early replication would have been pretty slow and crucially error-prone. Lots of mistakes.
But wait, maybe those mistakes weren't entirely bad back then. Could they have actually driven evolution?
That's exactly the idea. A high error rate creates variation, different RNA sequences.
And some of those sequences, just by chance, might be a bit more stable or better at copying themselves.
Precisely. Natural selection, but operating at the molecular level. The RNAs that were better at surviving or replicating would become more common.
So you get this gradual improvement driven by errors in selection.
And over time, maybe you get longer, more complex RNAs with better catalytic abilities, perhaps things that started to resemble components of modern ribosomes. These self-replicating functional RNAs are what our guide terms protogenomes.
Protogenomes, okay. And then the next step towards actual cells must have been getting a container, a membrane.
Yes, compartmentalization. Our material suggests these protogenomes might have started dabbling in basic energy metabolism, maybe using ATP or TTP, which are ribonucleotides themselves, conveniently.
Okay.
And then eventually getting enclosed within simple lipid membranes. That creates a contained environment, concentrates the molecules, makes everything much more efficient. The first primitive protocells.
Okay, so we've built this plausible RNA world in protocells. But life today uses proteins as the main workhorses and DNA for the master blueprint. Why the shift?
Good question. Our guide points to the sheer versatility of proteins. Because of their complex 3D structures, they can be much, much better catalysts than most ribozymes. More efficient, more specific.
So selection pressure would favor using proteins for catalysis.
It seems very likely. Over time, protein enzymes probably took over most of the catalytic jobs from ribozymes. And maybe, as our sources hint, once you're inside a membrane, evolving proteins that can interact with that membrane becomes advantageous too.
And RNA's role then shifts more towards just carrying the code for those proteins.
Primarily, yes. The protogenomes become templates dictating the amino acid sequences for the new protein catalysts. Now, there's some debate, mentioned in our material, about whether the original ribozymes evolved this coding ability or if new coding RNAs popped up. The latter is maybe considered more probable.
Okay. And then the big switch to DNA as the main information archive. Why DNA? What's so great about it?
Stability, stability, stability. Our guide emphasizes this. DNA is just chemically tougher than RNA. The deoxyribose sugar is less reactive than ribose. Replacing uracil with hymine also adds stability.
And the double helix.
That's the other huge advantage. Being double-stranded means you have a backup copy. If one strand gets damaged, the cell can use the other strand as a template to repair it accurately. Much harder with single-stranded RNA.
Right, a built-in error correction system. That's a game changer for long-term information storage.
Absolutely. So the plausible steps outlined in our guide likely involved evolving enzymes to make DNA building blocks, deoxyribonucleotides, from RNA ones, and then a reverse transcriptase-like enzyme, something that could copy RNA info into DNA.
So DNA takes over as the primary genome, And then these early DNA genomes started getting organized.
Seems likely. Our sources suggest they might have started as, maybe separate little DNA molecules, each coding for a gene. But linking these genes together onto larger structures, the first chromosomes would make cell division much more organized and reliable, ensures the daughter cells get the full set of instructions.
And this linking might have even started before the full switch to DNA.
Possibly, yeah. The organization could have begun evolving even while RNA was still heavily involved.
Now, thinking back to those building blocks forming spontaneously,
Ugh.
does that mean life could have started multiple times, independently?
That's a really interesting question our guide tackles. Given that the chemistry seems plausible, maybe multiple proto-life experiments were happening, but all known life today shares incredibly fundamental features. The same genetic code, basically, the same ribosome machinery, the same use of ATP for energy. This strongly suggests we all descend from a single common ancestral population, LUCA, the last universal common ancestor.
So one lineage just won out,
Yeah.
outcompeted any potential rivals.
That's the inference, yeah. The DNA-RNA protein system, combining stable storage, a flexible messenger, and highly efficient protein catalysts, was probably just far superior.
More stable, faster replication, better at grabbing resources.
Exactly. Our material speculates these cells might have even consumed any lingering RNA world organisms. The switch to DNA and proteins likely provided a massive competitive edge, leading to the dominance of this particular lineage.
It really makes you wonder, though. Are DNA and RNA the only way? Could life elsewhere, or maybe even hidden here, use a totally different chemical system for genetics?
That's a fantastic question, and our guide actually touches on this. It mentions research where synthetic chemists have made alternative base pairs that can function, and even some natural enzymes can work with non-standard bases.
So the machinery isn't totally rigid.
It shows there's some flexibility, and scientists have synthesized other types of nucleic acids altogether, with different backbones instead of the usual sugar phosphate.
Like those ones I mentioned, PNA, TNA, GNA,
Mm-hmm.
even that PNA with a peptide backbone.
Exactly. Threose nucleic acid, peptide nucleic acid, things like that. They can form stable double helices, sometimes even more stable than DNA, and can pair with DNA and RNA.
So what does that imply?
It implies that the chemical possibilities for storing genetic information might be much broader than just the DNA-RNA system we know. Maybe alternatives existed on early Earth but lost out, or maybe they're the basis for life on other planets with different chemistry. It opens up possibilities.
Okay, so from those deep molecular origins, life starts to take physical form. Let's shift gears a bit to the evolution of morphology, the shapes and structures of life. Our guide gives a kind of whirlwind tour.
Yeah, it starts with the earliest clear bacterial cells, prokaryotes, around 3.4 billion years ago. Simple but alive.
Then a huge leap, eukaryotes.
Yeah.
Cells with a nucleus, much more complex inside.
The chemical clues, like sterols, push their origins back to maybe 2.7 billion years ago, but the oldest definite fossil we have is Bangiomorpha, a red alga, about 1.2 billion years old. Its complexity suggests simpler eukaryotes were around even earlier, though.
And then things really start picking up speed, relatively speaking. Multicellular animals appearing around 640 million years ago.
Followed by the famous Cambrian explosion around 530 million years ago, this burst of diversification, where most major animal groups appear in the fossil record relatively quickly, though was followed by a big extinction event, too.
And then life conquers land. First plants, insects, vertebrate animals establishing themselves around 350 million years ago.
Then the age of dinosaurs, their extinction around 65 million years ago. And finally, our own ancestors, the first hominins, showing up really recently, geologically speaking, maybe 4.5 million years ago.
The big picture trend, as our material emphasizes, is this incredible increase in complexity and diversity over these vast stretches of time.
Absolutely.
And presumably, all these changes in body plans and forms are ultimately rooted in changes happening within the genomes.
That's the connection, yes. Our guide notes a general trend. More complex organisms often have larger genomes with more genes. Think bacteria with maybe under the 1,000 genes versus humans with around 20,000. Though we need to be careful not to equate evolution with just progress towards complexity. It's more nuanced.
Right, so if early life had smaller genomes, how did they grow? How did genomes get bigger and fancier? Our sources point to two main drivers.
Gene duplication and lateral gene transfer.
Okay, let's tackle gene duplication first. Sounds like a really important engine for evolution. How does it start?
Well, the simplest starting point is just getting an identical extra copy of an existing gene. Now you have two.
And the original function is usually still needed, right? So one copy has to keep doing the old job.
Generally, yes. There's selective pressure to maintain the original function in at least one copy, but the second copy, that's where things get interesting. It's redundant at first.
A spare part? What can happen to it?
Our material lays out the possibilities. Sometimes just having more protein product from two copies is beneficial, a dosage benefit, but often the spare copy is free to accumulate random mutations because losing its function isn't immediately harmful.
And most of the time, those mutations just break it, right?
Yeah.
It becomes a non-functional pseudogene. Our guide says that's the most common fate.
It is. But sometimes, and this is rare but incredibly important, a mutation gives that duplicate copy a new useful function. That's neofunctionalization. Selection then preserves this new version.
Creating novelty from redundancy. Cool.
There's also subfunctionalization, where maybe the two copies each lose part of the original function or become specialized for different tissues or times. So now you need both copies to do the full job the ancestor did.
And we actually see the results of this all over genomes, don't we? These gene families mentioned in our guide, groups of related genes that clearly arose from duplication events, like the globin genes for oxygen transport or the Hox genes for body plans.
Exactly. And some families, like the ones coding for ribosomal RNA, exist in many, many nearly identical copies. Our sources mention this phenomenon called concerted evolution, how these families manage to evolve together and stay so similar.
How does that work? It involves something called gene conversion.
Yeah, basically, during recombination, the shuffling of genetic material, the DNA sequence of one gene copy can sometimes get copied over onto the other copy. This homogenizes the family members. It can also help spread beneficial mutations throughout the family quickly.
Let's look at the globins as a case study, like our guide suggests.
Ah.
It's a classic example of neofunctionalization, right? They all bind oxygen or related molecules, but do slightly different jobs.
Absolutely. And using the molecular clock idea, comparing sequence differences to estimate divergence times, we can trace the history. An ancestral globin duplicated maybe 800 million years ago.
Giving rise to neuroglobin and the ancestor of the others.
Right. Then another split around 550 million years ago separates myoglobin in muscles from the blood hemoglobins. Then another split about 450 million years ago gives rise to the alpha globin and beta globin families.
And within those families, even more recent duplications created different versions for embryos, fetuses, and adults, fine-tuning oxygen delivery.
Precisely. It's a beautiful illustration of how duplication and divergence builds complexity over huge timescales.
And the Hox genes, those master control genes for laying out the body plan.
They tell a similar story. Vertebrates typically have 4 clusters of Hox genes, whereas insects, like fruit flies, have just one main cluster. This strongly suggests 2 rounds of whole genome duplication happen very early in vertebrate evolution.
Doubling the entire gene set. Twice. That must have provided massive raw material for evolving more complex bodies.
That's the hypothesis, yes. It likely played a big role. And some groups, like teleost fish, have had more duplications, ending up with 7 or 8 Hox clusters. Salmonids have even more. This might be linked to their own bursts of diversification.
Okay, so we see the results. But how do these duplications physically happen in the DNA? Our guide mentions things like unequal crossing over.
Yeah, during meiosis, when chromosomes pair up and exchange bits, if they misalign slightly, especially in regions with repetitive DNA, one chromosome can end up with the duplication of a segment, while the other gets a deletion.
And similar things can happen between sister chromatids.
Or even errors during DNA replication itself, like replication slippage, especially in repetitive areas, can lead to amplification or duplication of small regions. So there are several molecular mechanisms that can generate these extra gene copies.
And then there's that other mechanism mentioned, retrotransposition, which can create copies in totally different places.
Right. This is fascinating. A gene gets transcribed into mRNA, like usual, but then an enzyme called reverse transcriptase, often borrowed from retroviruses or endogenous retrotransposons, copies that mRNA back into DNA.
A cDNA copy.
Exactly. And this cDNA copy can then get inserted pretty much randomly back into the genome.
But these copies usually don't work, do they? Processed pseudogenes.
Often, yes. They usually lack the promoter sequences needed to be turned on, and they're missing introns because they were made from processed mRNA. But occasionally, one might land downstream of an existing promoter it can hijack.
So it becomes a functional retrogene.
Yes, often with different regulatory controls than the original parent gene because it's in a new neighborhood and lacks introns. And our guide notes we have hundreds of orphan retrogenes in our genome, where the retrogene is functional, but the original parent gene has actually been lost over time.
Wow. And sometimes duplication isn't about new functions, but just needing more product, like the RNA genes.
Exactly. Cells need vast numbers of ribosomes, so having hundreds or in some plants thousands of copies of the RNA genes ensures you can produce enough ribosomal RNA quickly. And concerted evolution via gene conversion keeps all these copies functional and similar.
Okay, so we've duplicated whole genes, but evolution also works on a smaller scale, right? Shuffling parts of genes, exons.
Yes, exon shuffling. Our material points out that many proteins are modular, built from distinct functional or structural units called domains. And in eukaryotes, these domains often correspond nicely to exons, the coding parts of genes separated by introns.
So you can duplicate and rearrange these exons like Lego bricks to build new genes with novel combinations of existing domains.
That's the idea. It's a powerful way to innovate by recombining pre-existing functional units. Our guide gives examples like the repetitive domains in collagen or tissue plasminogen activator, TPA, which looks like a mosaic assembled from domains found in fibronectin, EGF, and other proteins. It clearly shows assembly from parts of different ancestral genes.
Okay, that covers creating novelty from within. What about the other major route for getting new genes, acquiring them from other species? Lateral gene transfer or LGT?
Right. This is basically inheriting genes sideways, not vertically from parent to offspring. And as our guide emphasizes, LGT is rampant in prokaryotes, bacteria, and archaea. It's a huge driver of their evolution.
For allowing them to adapt super quickly, like sharing antibiotic resistance genes.
Exactly. It's a major reason antibiotic resistance can spread so fast. It really blurs the lines between bacterial species sometimes because they're constantly swapping genes.
Then how do they do it? Our guide lists a few ways.
Transformation, just picking up naked DNA from their environment. Conjugation, direct cell-to-cell transfer, often via plasmids. And transduction, where bacterial viruses, phages, accidentally carry bacterial DNA from one host to another.
Makes sense for single-celled organisms. What about LGT in eukaryotes? Like us, our guide says it's much rarer.
Much rarer, yeah. A key reason is the separation between germline cells, sperm and egg, and the rest of the body's somatic cells. For a transferred gene to be inherited, it needs to get into the germline.
Harder to do.
Much harder. But it does happen. In plants, allopolyploidy is a major mechanism hybridization between species, followed by genome duplication, basically smashes two genomes together. In animals, it's more limited, but there's evidence it can occur, maybe via retroviruses or transposons that manage to jump species barriers and integrate into the germline DNA.
Okay, so LGT is a big deal, especially for prokaryotes. Now, let's zoom back out to the whole genome level again. We mentioned whole genome duplication earlier in vertebrates. Let's dig into that a bit more. Polyploidy.
Right, copying the entire set of chromosomes. As our guide explains, this usually happens because of errors in meiosis, the cell division that makes sperm and egg. You might get diploid gametes instead of haploid ones.
And if two of those diploid gametes fuse,
you get a tetraploid, 4 sets of chromosomes. Sometimes these can be fertile, establishing a new polyploid lineage. Fusion of a diploid and a normal haploid gamete usually results in a triploid, which is often sterile, but tetraploids can work.
the immediate result is
For
just
everything, right?
double the genes. Massive redundancy.
Exactly. And that massive redundancy is the evolutionary gold mine. It gives duplicated genes enormous freedom to mutate, diverge, take on new functions, neofunctionalization, or divide up old ones, subfunctionalization, on a genome-wide scale.
So WGD events are thought to fuel major evolutionary leaps, big increases in complexity.
That's the idea. They provide a huge amount of raw material for innovation all at once.
And how do we know these happened, like hundreds of millions of years ago? Our guide mentions molecular clocks and synteny.
Right. Molecular clocks help if you see lots and lots of duplicated gene pairs that all seem to have diverged around the same time. That points to a single, large-scale event. Synteny is about gene order finding large blocks of the genome that are duplicated, often on different chromosomes, but still containing the same genes in the same relative order. That conserved order is strong evidence of a shared ancestral duplication.
And we see this signature in yeast, lots of plants, and the 2R hypothesis for those 2 rounds early in vertebrate evolution, plus specific rounds later in groups like fish.
Precisely. These ancient echoes are still detectable in genome structures today.
But genome-level duplications aren't just ancient history, are they? Our guide mentions more recent stuff in the human genome segmental duplications. or low-copy repeats.
Yes, SDs or LCRs. These are large chunks of DNA, anywhere from 1,000 bases up to hundreds of thousands that have been duplicated relatively recently. They're usually over 90–95% identical and present in just a few, maybe up to 50 or so copies.
And these are important.
Very. They're hotspots for genomic instability. Because they're so similar and repeated, they can misalign during recombination, leading to deletions, further duplications, inversions. This underlies quite a few human genetic disorders.
But they're also fuel for evolution.
Absolutely. They provide the raw material for new genes and gene families to evolve rapidly. Lots of genes involved in immunity and also some primate-specific traits seem to reside in these duplicated regions. And interestingly, our guide notes that the human genome seems to have a higher proportion of these large, very similar SDs compared to, say, the mouse genome.
Suggesting a burst of this activity more recently in our primate lineage.
That's what it suggests, yeah, contributing to ongoing evolution.
Okay, we've spent a lot of time on genes and duplicated bits, but most of our genome, like 98.5%, is non-coding, right? What about the evolution of all that stuff?
That's crucial. Our guide points out that generally non-coding DNA evolves faster because there's often less direct selective pressure against mutations. Changes can accumulate more readily.
But non-coding doesn't mean junk.
Right. Absolutely not. That's a huge misconception. Much of this non-coding DNA has critical functions. Regulatory sequences like promoters and enhancers that control when and where genes are turned on. Genes for functional RNAs like ribosomal RNA, transfer RNA, small nuclear RNAs, microRNAs, structural elements like centromeres and telomeres. These functional non-coding regions are under selection, often strong selection, and evolve more slowly.
Okay. And two major components of non-coding DNA with really interesting evolutionary stories are introns and transposons. Let's start with transposons, those jumping genes.
Transposable elements, or TEs. These are DNA sequences that literally have the ability to move around the genome. And our guide highlights their massive impact on evolution.
How so?
Well, they can cause mutations just by inserting themselves into the middle of a gene, disrupting it. Because there are often many copies of the same TE sequence scattered around, they can trigger large-scale rearrangements, deletions, duplications, inversions, translocations through unequal recombination between copies at different locations. We even saw an example earlier where line 1 transposon activity was implicated in duplicating the beta-globin gene region.
Wow. So they can really scramble the genome.
They can. Some TEs, like line 1s, can even accidentally carry adjacent chunks of genomic DNA along with them when they move a process called 3-transduction. They can also land near genes and alter their regulation, maybe introducing new regulatory elements or disrupting old ones. And there's even speculation they might facilitate exon shuffling by providing recombination points within introns.
So they're like agents of chaos, but also potential agents of evolutionary creativity.
That's a good way to think about it. They're a major source of genomic variation and change.
Okay, and the other big non-coding puzzle, introns. Those sequences within eukaryotic genes that get spliced out of the messenger RNA, they're mostly absent in prokaryotes. Where did they come from?
It's a really long-standing debate, and our material outlines the two main hypotheses, introns early versus introns late.
Okay, introns early.
This idea suggests introns were present way back, maybe even the last universal common ancestor, or potentially originating in the RNA world itself. Then, prokaryotes, with pressure for smaller, streamlined genomes, lost most or all of them.
And introns late.
This proposes that introns only arose later, specifically invading the genes of eukaryotes after they split off from prokaryotes. Perhaps they originated from self-splicing mobile elements, like group 2 introns found in some bacteria and organelles, that inserted themselves into genes.
And there's that exon theory of genes related to introns early.
Right. This model suggests that the very first genes might have been short sequences coding for single protein domains, like little minigenes. These got linked together, and the intervening DNA became the first introns. Splicing then evolved as a mechanism to remove these intervening sequences from the RNA transcript to produce a functional protein code.
And that connects intron origins to the whole idea of protein modularity and exon shuffling.
Exactly. It provides a neat potential explanation for why exons often correspond to protein domains. But, as our guide concludes, the debate isn't settled. There's evidence that seems to support both early origins for some introns and later insertions for others, possibly from TEs. It's likely introns have multiple origins and a really complex evolutionary history.
It's just staggering. Thinking about this whole journey From the Big Bang, the first molecules tentatively replicating in some primordial ocean, the RNA world giving way to DNA and proteins, the first cells.
Then the explosion of forms, the shaping of bodies through gene duplication, shuffling exons, borrowing genes, even duplicating entire genomes, all happening over billions of years.
We've covered so much the RNA world's potential start, the sheer power of gene duplication as an engine of novelty, the dynamic, sometimes disruptive influence of mobile elements like transposons, and just how fluid and changeable genomes really are.
It paints a picture of life not as static, but as this constantly evolving dynamic molecular system.
Absolutely. And thinking about it all, especially those alternative genetic chemistries we touched on,
Yeah,
it really makes you wonder, doesn't it? If life here took this drRNA path, driven by the specific conditions of early Earth, what else might be out there?
exactly. Considering the vastness of time, the potential chemical diversity in the universe, could there be life based on TNA or PNA or something we haven't even conceived of? Maybe even hidden away in some extreme environment right here on Earth.
It leaves you pondering the sheer creativity of molecular evolution. What other solutions to storing and replicating information might nature have come up with? It's a pretty profound thought to end on.
It certainly is. The possibilities seem almost limitless.